how mRNA vaccines have injected themselves into the world of medicine. Over 55 billion people across the globe have been vaccinated by the coronavirus vaccine, including everyone in this room right now. But have you ever thought about the work and development that has gone into preparing this vaccine for immunization? Throughout centuries of endless scientific research and development, a new evolution of vaccination arises as scientists are currently using a genetic sequence to battle pathogens in vaccines known as mRNA. Messenger RNA, mRNA vaccines, being both safe and efficient in production, are the alternative scientists have developed for a current preventative treatment. To further understand the development of mRNA vaccines, one must familiarize themselves with the function and process of mRNA vaccines, the advantages and disadvantages, the historical scientific development of such vaccines, and a further dive into the specific coronavirus mRNA vaccine.
mRNA vaccines have become extremely prevalent and are still being modified and tested today. The basic process in which these vaccinations function include the delivery of mRNA, intracellular trafficking, translation, and an antigen signal. The delivery of mRNA. This is where mRNA is encapsulated within a lipid nanoparticle, typically cationic and ionizable, and consisting of cholesterol, phospholipids, and polyethylene glycolipids. The initial attempt of delivering naked mRNA proved to be unsuccessful in transinfection considering the human body's natural temperature and pH levels. Thus, the nanoparticle structure was proposed, allowing scientists to successfully deliver mRNA protected by enzymic degradation and with increased stability and intracellular uptake. The cationic lipid promotes ionization at a lower pH for electrostatic complexation of negative mRNA molecules, thus allowing these lipids to be neutral at a physiological pH. Additionally, pH-sensitive lipids, or helper lipids, influence structural arrangement of mRNA lipid nanoparticles to improve factors of stability and intracellular uptake for cytosolic entry of mRNA. Once mRNA has successfully been delivered to a cell, intracellular trafficking occurs. This includes the mRNA lipid nanoparticle undergoing endocytosis with the host cell, becoming an endosome, and an endosomal escape. Consequently, translation then occurs. Ribosomes bound to the mRNA, moving left to right, and protein synthesis begins. Transfer RNA brings amino acids to corresponding codons, binding together via a peptide chain. Thus, a polypeptide is formed, before it begins folding to form the desired protein, which corresponds to the viral protein on the outer membrane of a virus. Considering mRNA vaccines are generally self-amplifying and based on an alpha virus genome, this results in antigen-encoding mRNA to be amplified in the host cell, mimicking a viral infection and sustaining levels of the spike protein to initiate an antigen signal. The antigen signal subsequently occurs when the protein is transported by the Golgi body to the exterior of the cell, where immune cells may identify it as foreign and begin forming specialized proteins, known as antibodies, to rid of the virus. Similarly, after the protein has been made, it may also be broken down within the cell by the proteasome into peptides. Thus, the body becomes familiar with the viral protein and, if contracted in the future, is more effectively and efficiently able to combat the virus prior to excessive infection.
mRNA vaccines have proved to hold multiple advantages and benefits over traditional vaccines that makes the vaccination a more reliable form of treatment. Hence, mRNA vaccines have proven to be relatively inexpensive, mostly seen in the mRNA coronavirus vaccine that costs less than other vaccines to manufacture. With mRNA vaccines being highly potent and versatile, they have the ability to execute their function exceptionally well and, when manipulated, for a wide range of infectious diseases during immunization. Due to the extensive and concise research that has been invested in creating mRNA vaccines, the known components of the drug allow the manufacturing and development process to be generally more efficient than conventional vaccines that can take years to fully develop as a functional pharmaceutical. Furthermore, mRNA vaccines are the safest option of vaccination due to the exclusion of using any live attenuated and inactive pathogens, thus eliminating endotoxins and infection.
Despite the numerous advantages associated with mRNA vaccines, there are still many existing flaws that are very disadvantageous, such as inadequate testing, the instability of mRNA, and the vaccine's low durability. Typically, vaccinations must be tested for 10 years to account for the short and long-term effects before authorization, with the FDA also being reluctant to perform in-human testing, and having performed mostly animal testing, there is minimal data and research that can depict long-term effects of mRNA vaccines in humans. Furthermore, due to the instability of mRNA molecule itself, it is quite difficult for scientists to produce a viable mRNA molecule for inoculation before its likely degradation. Additionally, due to mRNA vaccines being a relatively new advancement in immunization, long-term effects are also considered and unknown, such as longevity of immunity and whether it causes side effects such as increased inflammatory responses like redness or swelling, or, in the worst cases, aggravates disease.
Now that we have acquired all the specifics on mRNA vaccines, we can uncover the history that resulted in the end product. Scientific advancements in RNA vaccines date as far back as 1978, when scientists injected liposome-coated mRNA into mouse and human cells to induce protein expression and deliver genetic material into cells. Although this experiment was aimed solely at studying gene function, in 1987, the scientists involved discovered that synthetic RNA could function as an expression blocker of target genes in order to treat disease. In 1993, a team led by scientist Pierre Moulin was the first to attest that an mRNA in a liposome could promote a specific antiviral immune response in mice. In the late 1990s, Lipchin Technologies discovered that in order to prevent the mRNA from degrading, the mRNA must be coated with lipid nanoparticles, or LNPs, thus driving the opportunities for LNP delivery system in mRNA-based medicine. In 2015, Moderna expanded on this work and raised more than $1 billion on the assurance of using mRNA for vaccine production. Which leads us to the current COVID-19 pandemic, where biotech partnered with Pfizer and clinical trials began rapidly, starting at first in-human testing to emergency approval in less than 8 months, thus representing the steady but effective historical development of mRNA vaccines, which shapes society's understanding of current medicine today.
Now let's take a look at COVID-19 specifically. The coronavirus pandemic in late 2019 took the world by surprise, infecting millions of the world's population at a rapid pace. Thankfully, due to the pharmaceutical industry having studied and developed mRNA vaccines, companies like Moderna, Pfizer, and BioNTech began synthesizing mRNA that could be used in a vaccine to immune oneself of the coronavirus. The COVID-19 mRNA vaccine functions by containing a sample of the SARS-CoV-2 spike protein found on the virus's outer membrane. This includes the S1 and S2 protein, which binds to the ACE2 receptor. The mRNA vaccine is inoculated into the human body through the arm. The immune system will then read the mRNA as a set of instructions and begin replication. The body detects the replication of the foreign spike protein, producing antibodies that will destroy them and creating memory cells. Such cells will institute an immediate immune response if one is infected with the actual coronavirus before the virus is able to spread and inflict COVID-19 symptoms. As a result, the virus is eradicated from the body in a matter of days.
Conclusively, mRNA vaccines have proven to be an innovative step to a safer and more efficient form of immunization, in which scientists alike have developed genetic sequences to battle pathogens and diseases. Although the use of mRNA vaccines include disadvantages and implications, the level of advantages such vaccines provide further outweighs the negative. This is as it has a deeper focus on the preservation and protection of the human body, whilst also remaining effective and efficient in forming immunization in previous and uprising infectious and non-infectious diseases. Throughout the course of understanding the function and development of mRNA vaccines, the decades of historical and current research that led to today's mRNA vaccines, and the success rate of the vaccine used to immunize the global population from COVID-19, one can deduce that mRNA vaccines are a positive headway in the scientific development of pharmaceuticals, therefore providing the world with a trustworthy and a viable source of vaccination.